to be presenting here at the University of Chicago. So just to start off, we'll do a quick refresher on how Nanopore sequencing works. I know it's a self-service core. Have some of you had an opportunity to use the instrument? Yeah, okay, nice. So this will be a refresher for some of you. So the core of our technology, right, is this biological nanopore. You can see a cross-section of it here. So these naturally exist, right? They act as a gateway between two different regions, typically in a cell. So we have mutated the nanopore to sort of suit our needs in terms of using it to produce biological sequence data. And so some of the key mutations are the length of the overall pore, the size of the barrel, and then this in the cross section, this sort of bluish part, that's like what we call the choke points, and I'll go into the purpose of that. So here's a little fun video. So what you can see is that the nanopore is embedded in this supporting membrane. That membrane is electrically resistant, and these little gray, like wiggly worms that you can see reaching up, that's a polyphilic molecule that we introduce to the flow cell during loading that is attracted to the actual sequencing library and sort of pulls it down to the surface of the flow cell to encourage docking. The other key components here are the motor enzyme. That's literally a helicase. It's going to act as a molecular ratchet. It will unwind the double-stranded DNA or the RNA cDNA complex. And it will also feed the DNA through the pore at a controlled speed. So since it's a helicase, it burns an ATP to move one base, and it moves 400 bases per second. It's really important that we control that speed because that's what's going to allow our electrical signal that we're generating to be legible. The way that we're generating an electrical signal is that this pore is embedded in the electrically resistant membrane. And then while we're translocating the DNA through, we are also flowing a salty buffer through there. So ions are passing through the channel. And each base that passes through is going to disrupt that flow of ions, creating that characteristic pattern. And the canonical base isn't just going to create a characteristic disruption, but also any chemical moiety that sits on top of that base. So that's what allows us to call 5MC, 5HMC, and then a whole suite of other modifications, especially in direct RNA, just from sequencing a native molecule. The way that we actually take this electrical signal, which we colloquially call the squiggle, the way that we actually turn that into a base called FASTQ is by using a machine learning or AI model. We've traditionally called it machine learning. We have benefited from like the AI revolution in that some of our most modern and cutting edge base calling models are a little bit informed by like the structures of like the modern complex learning language models. Like we use some transformer models. So these are very large, very complex, but they have allowed us to get a significant accuracy boost. So that's been kind of an interesting cross section. So as you are probably aware, the base calling component of this relies on GPU power. Fatigue has a very powerful GPU. It's a 4090, is that right? It's a good one. So you are well outfitted at your core. And then we have several sort of benefits to sequencing this way. So that direct sequencing component that I mentioned, that allows us to capture methylation information without having to do a bisulfite prep. You actually don't have to do any kind of additional prep to capture methylation information. We generally do either a classic D-A-tailing prep, or we can do a transposase-based prep. And again, as long as we're not sequencing something that's been PCR'd, we're able to capture all sorts of epigenetic information. Avoiding PCR allows us to, not have to worry about amplification bias. That said, we can absolutely sequence PCR product. That's actually something the pores like to sequence because it's been nicely prepared. There's no nicks or gaps. You'll notice in the library prep protocols that we have you use that FFPE repair enzyme, even when you're not sequencing FFPE. And that's just because when we are sequencing native DNA, especially long native DNA, it's very helpful to have the strands put through like a conditioning and repair process. Otherwise, the helicase molecule, as it encounters like nicks or gaps in the DNA will start to sort of slip. So you can see the strands actually break off, and you'll see an artificial truncation of your sequencing lengths, your average sequencing lengths, or you can even see like some quality problems if your DNA is in rough shape. Sort of like any other, feel free to grab a treat on your way in. So that's one of the ways that like, I mean, obviously quality of your input impacts sequencing no matter what platform you're on, right? But that is like an important thing to think about when you're considering native sequencing, and especially native sequencing if your priority is getting something really long. Our longest read lengths are going to come from really fresh stuff, but then also doing everything we can to make sure the DNA is in the best shape possible will contribute to long read lengths. And then, you know, if that's not possible, we do always have PCR options. Obviously, we're known a little bit more throughout the industry as a long read platform, along with our friends at PacBio, but we are in fact read length agnostic. We can go down to 50 base pairs. We had a flirtation with 20 base pairs, and we're exploring that, but we decided to sort of tighten things up. We know we're really good in the 50 to on the upper end. The largest molecule we've ever called was 4 megabases. So we know we're really good in that range. But we want to push the lower limit as well. So that's one thing that's really useful. You might ask yourself, why would I use the Nanopore sequencer to sequence something that small? And a couple of the answers are, we have significant interest in sequencing small RNAs, which is a whole world in of itself, right? Because some of those RNAs have really wiggly, annoying shapes that are hard to get through a pore, but it still is a cool thing to look into. And then the other answer is we do have significant interest in cell-free DNA size stuff, and cell-free DNA methylation is kind of an interesting up and coming area of pursuits. And then we do have the option for real-time analysis. This mostly comes up, so because we have no bounded cycle threshold, the Nanopore platform is very flexible, right? So I was an Illumina user for 7 years before I came to Nanopore. And you have to complete your 300 cycles or your 150 cycles, right? With Nanopore, we're just running based on time. We have flooded our Nanopore array with DNA, and we are letting our molecules go through our pores, and we're essentially running until we get as much data as we want, or until we've denatured all of our pores. Since it is a biological nanopore, the pores don't last forever, right? It's a little bit like a battery that has a limited lifespan. So we can run down the battery until we've used all the juice, or we can run it in bursts and wash the flow cell and then use it again. But this does give us the ability, we can output chunks of data essentially just based on like a JSON file or just a setting in Minnow. So this is mostly, the most times that I see this applied is in like the molecular microbiology world, where people want to do like a live assessment of like metagenomics, or even like 16S sequencing. There's also some interesting applications for it in the space of live metagenomic assessment of brain cancers. So like resection of a tumor and then methylation profiling. They've done that actually like in like live during surgery, which is obviously very cutting edge and sounds a little crazy, but I think it's really interesting since, methylation profiles are very important to glioblastoma and can tell you what is and isn't eligible for treatment and what shouldn't be resected. And then of course there's this element of chemistry on bespoke electronics. One of our missions is to democratize sequencing, right? So the devices that you have in the core here are the Mark 1B, so the min-ion scale sequencer, and then the P2 Solo. So these are both devices that you can plug into your own computer. Sometimes, that's great, right, in terms of access. You guys are very lucky, and we at Nanopore are very lucky that Petit has invested in quite a high scale and very nice computer to go with that, so that you are getting like an experience that's close to what you would get with like an integrated device. Yeah, and that allows us again to have access. So the P2 Solo, it's 2 PromethION slots, and each PromethION flow cell can, I'll get to that in a minute, can produce like 100 gigabases of data, right? So it is a very powerful sequencer. And it's just plugged into a powerful desktop computer. Any questions about that? All right. So one thing that I want to just make sure you guys have this in mind. This is one of like the key features of flagship applications on Nanopore. This is our adaptive sampling technology. And when I describe it, it's going to sound fake, but I promise it's real. I get to look at real examples of data and experiments that use it. But what we do, because we have this ability, right, to live base call and also do live alignment, we set up an experiment where we've just prepped a normal library, right, generally via ligation sequencing. And then we feed the instrument's reference file for the genome. And then in addition to that, a bed file containing targets. What we do is then we have the instrument sequence, base call, and live align the read that it's sequenced, and then check against that bed file to see if the targets in the bed file are present in what it has sequenced so far. If the answer is yes, it will continue sequencing the strand. If the answer is no, it rejects the polarity across the membrane, or reverses the polarity across the membrane and rejects the strand out of the pore. And this way we are able to live enrich on the sequencer. So we can get like 5x to like 100x coverage over baseline, depending on how the experiment is designed and what kind of flow cell we're using. Some of the key aspects of this and like the important limitation is that this is sort of like I like to think of it as like a needle in a haystack, right? So if you have a pile, we want to have like a pile of needles and hay, right? So generally we want, if we're doing enrichment, to have our target be, we want it to represent about like 0.7 to 5% of the genome. So while we're accepting and rejecting, right, we do want to be frequently enough pulling out needles, right? Because if we're just spending the whole time finding hay and saying no, it's just not a very efficient sequencing process. But it works really well, even if we can hit that lower point of like 0.5, 0.7% of the genome. And sometimes if people have, maybe they only have one gene. I have a group that really just investigates the MHC complex. They look at HIV integration in human and primate genomes. And so we just have them target the whole chromosome arm. And using that strategy, they get enough data to actually do assemblies of the MHC region. So kind of interesting. Again, I know it sounds a little bit outlandish, but it does work quite well as long as we're kind of playing within the bounds. And then on the other end, right, when we go above, like the theoretical max is 10% of the human genome for enrichment. And that's just because if you can imagine, like if you just had a big, huge needle and like a couple pieces of hay sitting over the top, you wouldn't be worth it. Just sequence everything, right? Yes.
Hey, Maddie. One question about the adaptive sampling is how strict is it in terms of the sequence type? So if there's, whether it's an error or a variant that happens, is that you don't anticipate that most of the sequence lines up, will it still keep it, or will it fit it out?
So it's pretty loose in terms of like we would not be rejecting based on like a SNP, and I can get you like specific numbers, because we want all of this to take place really quickly, and we're relying on essentially, we're trusting the computer and we're optimizing the system to be able to handle a lot of computational tasks, right? So for this to be efficient, we need to be making this decision within like 2 to 3 seconds. So within like 400 to like 800 base pairs, right? So to do that, we first of all need like a good high quality computer, which you have. And then the other component is that we do run this in fast base calling, so our lowest stringency base calling. And then we do have, like, I can get you some more specific numbers about like what exactly like the mismatch tolerance is. But we would definitely not be rejecting based on like a SNP. It's quite loose.
Do you suggest then after collecting a sample this way, or the data this way, then running it back to the base caller with like the higher quality?
And we do have, so we have a couple different options here. We actually recently introduced a feature that balances the compute a little bit better, where we base call the on-target reads only. So sometimes people are like, why would you ever base call the rejected reads, right? But it's because a lot of times when you are running a targeted panel, you kind of rely on your off-target to like review your overall coverage of the genome, to, set some baseline metrics, right? So we wanted people to have the option to do that, or to purely focus on their accepted reads. And in that case, we can support running live base calling with our hack base caller, and then simultaneously adaptive sampling. It's always going to run a separate base caller purely for the accept reject decisions, and then anything layered on top of that is separate.
Just so you guys are aware, our high performance cluster here has several GPUs that are exactly the model that you guys got.
Oh, nice.
So we're able to do extensive base calling both things if we want.
Perfect. Nice. Yeah. We love to see that. Yeah, so we jumped ahead a little bit, but this is just like an idea of what I wanted you to take away from this essentially is that we have two different scales of flow cells, promethion and min-ion, and you have access to both here in your core. We do have the smaller flongle device. The fate of the flongle is a little bit unclear. If you need a flongle, you get to talk to Braun and do negotiations with him. Bring him a snack. Yes.
Can you put Silver Island? No.
There's a nice woman in Wisconsin who's always giving Braun jams. So if you have a jam, maybe you could get a flongle. No, I'm not clear on what the rule is. Do you know the rule, Braun? Can you get flongles?
Oh. It's not on the store. Well,
I mean, if it's really not supported, there's kind of.
there's just so.
I will say about the flongle, yeah. I mean, the flongle is so alluring for its price point, right? And that price point is so good because we make it so cheaply and unstably. So our plan is to make a stable version of the flongle that does not cause quite so many headaches, because we have seen, quite a few. The flongle is kind of like we envisioned it kind of like as a fun toy, like especially for like educational stuff. And I think we didn't appreciate how useful the niche of that data output would be, because it's easy to think like more data is better, right? But sometimes there's many applications where it's really useful to get, like to have a small data footprint and a very affordable flow cell. So it's definitely something that we're very aware of, and we're working hard on the replacement. I have seen the replacement. It looks like a vape. So it's kind of cute, kind of fun. So hopefully you will get to see our vape soon.
In defense of the flongle, I did use it in 2022 for a while.
Good. I'm glad you had a good experience. I mean, it's very, it's very dependent on, like, if you're sequencing amplicons,
Yep.
Hell yeah. If you are sequencing like a weird, like lichen that no one has ever sequenced before, that's going to be a tough one. But
I think other than
yes.
Also,
yeah, you're the ideal flongle customer.
which did get me because once you do it's a little.
I see.
Mm-hmm. This is just, I just wanted to give you guys a snapshot of where we are at with our accuracy right now. So again, I mentioned those like transformer models. Those are these 5.0 models. So these are very big and slow, but they are effective. So this is how we get like Q26. And then this is actually, I think, this like a touch behind actually, because I pretty regularly off of like a fresh like human blood extraction. Sorry to default for human. It's just common to sequence, right? I see a lot of like Q scores of like 23, 24 running hack off of human stuff. And then yeah, with the big soup models, you can get like 26, 27. And then some of like the fluctuations in this. Do we have any? So when I think UChicago, I don't really think agrobio, but is that wrong? Do you guys have plant people here? Maybe. Yeah. You have some? Okay. So we do absolutely train our models to include plant data. And we do have like some kind of esoteric bioinformatics tools that are really useful for gap filling and assembly applications in plants. But your kind of baseline read scores will be a little bit lower sequencing plants just because of their horrifying genomes. But it does still work. It's just like baseline. It's a little bit easier to speak to humans just because we have so much data on sequencing humans and then other mammals. And then, it gets weird when you get into like frogs and stuff. But generally, this is very in line with the performance that I see. I kind of hinted at, the nature of the pore being important to the way that the Nanopore quality work. So when we changed from I apologize for our love of numbers and like version coding, but we changed from our R9 nanopore to our R10 nanopore. And the key changes that we made that we're able to improve our accuracy to like a baseline modal Q20 are this elongation of the barrel and then this addition of a second choke point. So the longer barrel essentially allows us to get more context when we're sequencing through a homopolymer region. So, yes, as you can imagine, like with any sequencing platform, the most difficult thing for us to sequence through is homopolymers, a long stretch of homopolymers, right? So having this longer barrel that allows us to, like see more of what's on either side, it's kind of almost like interpreting like a SAT question. That's been really helpful. And then the choke points. So at its largest spot, the nanopore is like 10 nanometers across, and then it narrows down to like 1.6. And the choke points, it sounds a little scary, right? Like we're choking the DNA, but the real idea here is to get a really stringent and specific signal. So adding a second one just allows us to essentially do like a double check. So we're adding stringency and really narrowing in on the signal 2 times. So that's what boosted our accuracy, just to give you a peek behind the curtain. And this really did help with our homopolymer resolution and our performance in indels. Most long homopolymers and indels are found like outside of coding regions. So you'll see like a significant discrepancy here when you look at our indel benchmarking. These kind of double sets of bars, the solid bars, that's all indels. And then the longer, like extended bars, that's if you only look at within coding regions. And that's just because outside coding regions is where all the gnarly stuff tends to live. But this is an idea, just like at a glance, of our performance for general base calling, SNVs, indels, and SVs. Obviously, like the place where we really excel is structural variation calling, but we also have like some surprising advantages. There's some interesting papers showing that long reads, the superior mappability and assembly options for long reads really does help sometimes with SNP calling and can boost accuracy in that way. And then this is just a comparison of hack versus soup. We would like you to feel comfortable using hack for most applications. There are some applications where soup is useful. One of them is single cell because of the enhanced UMI detection. But in general, hack performs very well. And then when we're doing, we tend to talk about 30x coverage because we see really solid performance of human genome variant calling at 30x. I know like our colleagues at PacBio are kind of pioneering a 20x. We have some people experimenting with that on Nanopore as well. We're kind of sticking with the 30x benchmark, but of course we're happy to consult on that. And obviously it depends on like what you want to do, right? Like methylation applications, for example, it really depends on what you're looking at from the methylation data, how deep you want to go. This is just an example of how you get the mod call. So you just turn on the switch. And then you do have some different toggles. So we can do, everything's paired together in terms of 5MC and 5HMC. We can do CPG context or all context, and then we can do 4MC as well. Yeah, so you just switch this on. The all context callers are locked to super accuracy base calling, and that's just because they're big, huge callers. They need to be able to like really look throughout the genome. Obviously, 5MC, 5HMC, CPG is a lot more human focused, a lot more, yes. Methylation. Yes, we would like to, absolutely. We're always looking for stuff to add. A lot of it is informed by just like what we get requests for, what we see is like up and coming in the literature. But yeah, we're always taking suggestions. Okay. Your own. Okay. Yeah, absolutely. Yeah, let me ask about when that'll be incorporated for sure. I can get that data to you for sure.
Good question. Could you differentiate the 5MC and 5HMC?
Yes. Yeah, so they're paired together, like the caller just, it was easier for us to just have it call both simultaneously. If you're using Dorado command line, you can like subtract one out, or you can just ignore the one you don't want. We used to have them like listed like as individual callers, but it's computationally easier to package them together, but they produce differential calls. And so, sorry.
Oh, thanks. If you're gonna get to this at some point, then you're ready to answer now, but I'm still a little bit confused on how I should think about like sequencing depth. With Nanopore, like coverage, like how should I be thinking about it?
Yeah, like are you used to like an Illumina sort of
Yes.
mindset? Yeah. Yeah. I mean, so that's a great point. I know like a lot of times like Illumina, right, will just kind of tell you like this many reads like over
Exactly.
your target, right? And we can kind of say the same thing. The main translation aspect, in my opinion, from Nanopore to Illumina is just not thinking in terms of bounded reads. So
Sure.
a lot of times we speak in gigabase output.
Yeah.
And you
Right.
can kind of do the calculation if you
Right.
think about your region size, right? Like, so I want to cover like 1,500 bases. And then if you just think about how many times you want to see that, like I want 15. I mean, a lot of times it is sort of cross-compatible with some of the Illumina recommendations. A lot of times they're higher just because like they don't capture as much per read. But in general, like you can think like I want to see my 1,500 base region this many times, and then just think about how many gigabases that is, and then compare that to the gigabase output of your given flow cell.
Sure.
That's kind of how we recommend doing it. And for some applications, we try to provide like a calculator. It is hard for us just because we're so flexible and we have so many different applications, and it's all like provided by us. We have like a couple partnerships where people, have like a designed assay, like there's a carrier screening assay from Asuragen now
Okay.
that has like more defined boundaries, like it's amplicons. They're telling you like how many per flow cell and how many fit.
Sure.
But I'm also always happy to like kind of walk through that kind of thing.
Sure.
Of course. Yeah, so one thing that we know is that like approximately 8% of the human genome is just inaccessible to short reads because it can be really hard to map, your little 300 base pair puzzle pieces, right? It's a lot easier to put a puzzle together from big, huge pieces or even medium-sized pieces, right? And then we also have this additional layer of methylation information that we consider as a part of the genome that isn't captured by a traditional short read library unless you layer on like a methyltransferase assay or what do they call it, 5-base chemistry, or like a bisulfite experiment. So we feel that you get more information out of the Nanopore system. So here's just a list of what we have right now. So 5MC, 5HMC, 4MC, 5MC, 6MA are what we have for now. And then we are integrating 4MC, 5MC into Minnow soon. But everything else is in there. And then for RNA, we have 6MA, pseudouridine. We actually also have like methyl pseudouridine for biopharma, and then the 5MC and the inosine models. And then we do outperform bisulfite at this point. So when we are testing our methylation, we have to actually like design synthetic strands because we have exceeded the accuracy of bisulfite. So if we try to align our data to like a bisulfite data set, it will look like we are wrong when in fact the bisulfite data is slightly off. And then just another snapshot of kind of where we're at. So simplex, we briefly had a flirtation with duplex sequencing, right? Where you capture one side of the double-stranded molecule goes through, and then immediately after the other side goes through that same pore, and then you can pair them together bioinformatically. That did yield like a Q30 score, but it was a real difficult thing to achieve, partially because like if you're aware of all of the sort of patent drama around molecular hairpins and attaching two strands together. So that was hard to get around, and then it was hard to optimize. And then also you lose, you have to compress your data down, right? Your two sides of your strands, you have to compress everything down into one read. So you're losing a significant amount of data. And based on our testing, we felt like getting twice as much data at Q27 was just a way better deal than getting half as much data at Q30. There are still like some interesting potential applications for duplex, but if that's like something you heard about with Nanopore, for a minute we were really hot on that, but that's kind of why we've drifted away from it. It's still like in our, call it Nanopore skunks works. We're still working on it, but that's kind of where that went. And then we've gotten really good, particularly in like plasmid and bacterial assemblies. We're excellent at that. We've gotten some really good T to T nanopore only assemblies, which I know for a long time was like unthought of. People felt like you had to use Illumina, PacBio, and Nanopore. And then we're getting, you know, really good human variant calling, as well as our methylation performance. So to dive a little bit deeper into methylation, right, like I remember when I was in the lab running bisulfite assays, running microarrays, the creak of the beloved high boven. I mean, I felt like I was doing a lot, but also I wasn't having fun. So definitely something to consider when you can get canonical base calls and methylation information all from just one experiment. So we want to, you know, remind people that that is an option today and encourage people to, you know, consider Nanopore. Even now, like, because we have expanded our methylation caller offerings so much, and because you have access to the PromethION instrument, our library preps are really simple. This is highlighting, sorry, this is very marketing. The 10-minute library prep is like the rapid barcoding one. You'd probably use the ligation one, which takes about like 3 hours. But then this is compatible with some of this methylation stuff is compatible with some of like the epic array based methylation callers, and we can provide guidance on that. I do have, she is not present, but my beloved colleague Jennifer is a field bioinformatics scientist who works out of Marion City, Iowa, and she does come in to Chicago sometimes to help us. So if people want more information about bioinformatics, we're happy to bring Jennifer in to do one of these that just talks about analysis. So definitely let us know. Yeah, and then this is just a sort of a summary of what kind of data you would be getting from, you know, ONT whole genome sequencing with methylation switched on versus whole bisulfite sequencing versus RRMS is an adaptive sampling based version of RRBS. So you don't have to do any kind of bisulfite conversion. We just feed the instrument a bed file that contains the CPG islands of the genome, and it just enriches for those. We can do this multiplex on a PromethION instrument. We can do 5 samples on one PromethION flow cell, or we can do 1 sample on a MinION flow cell and get the essentially what you would get from RRBS, but a lot easier. And then of course you're getting way more regions either way compared to an epic array, or a chip as some call them. And then obviously when you combine methylation and long reads, you get into really cool phasing capabilities, right? So once we get into like 20 to 30 KB long reads, we start to be able to really, really clearly phase methylation. So this is an example where you see this like this differential methylation in like red and blue over this one locus. So this kind of thing is really useful, of course, in like imprinting disorders. We see this used a lot in rare diseases. There was an interesting paper recently that addressed this in like retinoblastoma, identifying like a parent of origin, and it even helped like determine if the variant that was suspected was pathogenic or not. So this is something that, you know, we deal with a lot when we're talking to people who are more in like the rare disease space, like on like verging on clinical, but you know, they're kind of more in like the clinical group couldn't get an answer. And so now we're kind of trying different things. Yeah, and this is one of the cool things that we like to see. Obviously you can load, we provide when you get your methylation calls, they come out as mod BAMs, and then you can convert them into bed methyls using like a toolkit that we provide, and then you can view them natively in IGV, or we have a bunch of different tools that allows you to process them. But this is kind of a different way of thinking about this compared to like epic arrays, so it's always something that we're happy to provide guidance on. And of course, super relevant for imprinting disorders. So to get into direct RNA a little bit, right? Lots of reasons to sequence RNA. Is anybody here more focused on like transcriptomics, that kind of thing, or just a little bit? Okay, nice, nice. So obviously we absolutely do transcriptomics. We like the normal way, the cDNA way, and we have a recent improvement where we've taken our read lengths from like an average of like 1 kb to like 2 to 3 kb, which is exciting because obviously we all want to sequence all the way through from the 3' to the 5' gap. But also it can be really interesting to sequence RNA directly. So this allows for the identification of RNA-based modifications. We can measure poly A tail lengths, and then we can, investigate alternative splicing. We can also do that with cDNA, but do this isoform mapping and then transcript quantitation as well, which you can also do with cDNA. So some disadvantages of cDNA, right? The main thing is just that you lose base modification, which historically people haven't been super worried about, but I know there are some groups here at the University of Chicago who specifically investigate RNA, which I think is pretty cool, RNA modifications. So to get into that, our first RNA modification caller was this M6A Dretch context versus all context caller. So here's our accuracy numbers. Our accuracy is a little bit lower for direct RNA. We have recently, this does represent like an improvement though. We have to switch to an RNA specific nanopore to improve the signal and get better signal to noise ratio. And that's been going really well. The other kind of component, this is an interesting library prep if you go through it. It's super easy to do, but you do generate, there's a cDNA generation step, which I know like freaks a lot of people out. But if you go through the protocol, we exclusively generate a single strand of cDNA, and it is not eligible for the attachment of our sequencing adapter. So only the RNA side gets the sequencing adapter, and the cDNA is just there to act as like a support and a scaffold as the RNA is translocated through the pore. So we do encourage doing that step just because it prevents the RNA from folding up into its superstructure and making it really hard to tug it through the pore. But yeah, this has been a really interesting sort of emerging field, RNA metagenomics. So the M6A is probably the most popular caller, but we do have some other ones. So pseudouridine, this one's been really hot and controversial on Twitter because it's in the vaccines, which actually this has been used at a lot of biopharmaceutical companies that we're talking to, where they want to make sure that their RNA that they're using for vaccines is thoroughly modified with the pseudouridine, and it has the cool pitchfork symbol. So this was a good one. And then inosine, I believe this one is implicated in certain like cancers. So I would like to see more information on inosine. As you can see, we're kind of starting to rack up our RNA modifications, and I feel like, I'm excited to see more data come in from these. And then also maybe I'm thinking of M5C is the cancer one. And then, of course, you can also get transcriptomics information, right? Your standard transcriptomics. One word of advice, if you are not interested in RNA modifications, I would point you towards a cDNA approach just because you get more reads. We have to translocate the RNA significantly slower. We use 130 bases per second speed, just because the RNA is fragile and we have to be careful feeding it through the pore. So if you are interested in gene counting, if you're just exploring alternative splicing, I would go for the cDNA approach, but I'm also happy to talk through that kind of thing in more detail. And this is an example of the advantage of getting a higher like transcriptome depth, and then just like the ability, right? It is really useful to be able to sequence a full length RNA read, right? And to be able to capture these like large transcripts instead of just being bound to like a 300 base pair fragment. And then we can measure poly A tail length. So this just comes from the natural sequencing process. So we do the library prep sort of anchors initially off of the poly A tail, and then we can do some fun math that allows us to estimate the size of your poly A tail length. This is another one that's really popular in biopharma. And also at like some interesting service centers, and they have pushed us, as you can imagine, to get increasingly good at it. So I know there's like some legacy technologies or orthogonal methods where you can estimate a poly A tail length, but it's cool that we can just do it on the sequencer now. And then we do go down to, for a long time, we could only do 200 base pairs or higher for RNA, but now we can go down to 50, which the RNA people, in my experience, are some of the most tenacious. They are always looking to sequence weirder and weirder and smaller RNAs. So, you know, the next boundary is probably 20 base pair RNAs, but I think this was an exciting improvement. And we were also getting, this also enabled us to sort of boost our overall yield. Yes, and so then to get into single cell. So obviously single cell kind of hit it big in 2018, Nature Method of the Year, and then, you know, around that time, long read was also Method of the Year. And so we think these are two great tastes that taste great together. And here's why. So you are just missing a lot, right? When you're doing short read transcriptomics. This is a little misleading because obviously you can do a random hexamer approach and grab fragments of RNA from all over your transcript region, but then it can be hard to like track them back to whether or not they were poly A-tailed, whether or not they're biologically relevant. So, you know, pros and cons, but we do feel confident that when you are doing long read transcriptomic sequencing, that you are much better able to capture, you know, the full transcript and to understand, you know, the full breadth and role of the transcript, and then to investigate things like alternative splicing and isoforms. So we're missing out when we're doing short read on all of this information. And there's really, you know, not a disadvantage from switching to long reads to get all of this additional advantage, and we'll hopefully be able to prove that. Sorry that this slide is a little complicated. The only thing you need to take away is that when we're doing, so our main example is the 10x cDNA platforms, just because we have a partnership with them, we can support other single cell, thank you, other single cell platforms, but 10x, we just have like a lot of benchmarking. We have a wonderful app note with them. But essentially, the 10x cDNA generation process uses a TSO-TSO, uses a template switching oligo, which does create a TSO-TSO artifact that is invisible on Illumina sequencers because it doesn't have their read one primer. It's super visible to us because we don't care about the read one primer, and we will just put our adapter onto it. So we have to do this quick little step at the beginning where we pull down the stuff that we really want and we wash away that TSO-TSO artifact so it doesn't just take up sequencing volume. Other than that, though, you just take your unsheared 10x cDNA. You can take cDNA from, you know, 10x experiments that you already have short read data on and just take them through long read to get more information. This is actually this slide looks scary, but this is actually an extremely simple library prep. It's just a DA tailing with a little fun step up front. And then we just put it onto the sequencer. And, you know, the things to think about are, you know, just how much are we missing out on when we are only doing the short read single cell work. So let's look at some examples. So this is a cool example. So what you can see on the left is isoform expression by Nanopore. This is actually taken directly from the 10x application note. So you always get these like t-SNE plots of gene expression, right? And the 10x application note actually shows that you can get the same data of a gene expression plot from Nanopore and Illumina, very equivalent performance, but then because you get the long reads on Nanopore, you can additionally create a t-SNE doing isoform expression exclusively. And then using this Visium data, this is an interesting case where we see a significant difference in isoform expression in a mouse brain. So there's like two different isoforms. So 202 is like not expressed at all in this mouse midbrain, but 201 is highly expressed. So information that would have been totally invisible on the short read sequencer, right? Just wouldn't be able to see it at all because it's isoform related. I think just my I think my screen is just not not calibrated well, but
Oh, sure it is.
yes. And then this is an example of like an age-related differentiated expression. So these precursor cells versus the mature cells are seen expressing two different isoforms. So the 206 is expressed in the immature cells, and then over on the other side, the mature cells and the precursors are showing the 204. And they found a bunch of different cases of this. This is another mouse example. So they show this isoform switching behavior throughout the development of the brain. So this is another cool one that would, again, we would not be able to see on short reads that the cells were switching during maturation. This is an example where we were able to essentially like assess the mutation volume throughout more of a tumor-based example. So obviously you would expect a tumor to have like a bolus of mutations, but it's interesting to see it right on the single cell level and mapped out like this compared to the wild type. So just like the emphasis here, right? The point that I'm making over and over again is that you can get all the same information you would get from a short read experiment, but then this additional entire other layer of information without sacrificing what you would get off of the short read experiment. So you can do what you would already do and more. Do I have one more? Okay. And then here's one more. So this is right, we have our Illumina regular abundance matrix, right, where the cells clearly differentiate into these two clusters, right, cluster 10 and cluster 2. But if we layer on top the splicing information, we can see that they further differentiate into these subclusters, and that this is all based around this intron retention event that's completely invisible on the Illumina sequencer because they just didn't capture any reads that go across this region. So this is another cool one where you just get a lot more detail and a new layer of understanding. Again, this is a promethion scale application. It's showing the big promethion. Sorry, that's marketing. You can do it on your small promethion, no problem. And then we have our own Epitome Labs workflow where you can process your single cell data, and this will be compatible with things like Seurat and other downstream applications, as well as like a whole host of like third-party tools. We can support fusion calling, we can support SNP calling, and then this is, a big emphasis on us. So we think it's cool. And I can also go into like, if you're interested in this, like numbers, how many cells you want to put in, how many reads you can expect to get out. And I know I've already kind of hyped it up, but I just really enjoy the 10x app note. It's just such a good resource. So it's good to check out. As you can see on the left, we're just kind of showing the equivalent performance of the Illumina and ONT libraries on the different platforms, just in terms of, like being able to identify UMIs, gene saturation, very similar performance. And then again, we're seeing Illumina and ONT, they overlay pretty much perfectly on the gene mapping. And then the isoform map only can be provided with Nanopore data or long read data. I will be generous to my friends at PacBio, they can do it too. But their library prep's a real headache. Yeah. Any questions? Sorry, how am I doing on time? Okay. I do want to give you guys time for questions. No questions.
Okay, there's a question.
Yeah. Oops, sorry, not that. Chat. 2 questions. Okay, so for adaptive sampling, that is a really good question. So if you think about the fact that we need to make the decision, right, within, we're translocating at 400 bases per second, and to be efficient, we ideally want to make the choice within, 800 bases. Otherwise, like we've sequenced a little bit too much molecule to be really efficient. So there are a couple papers that show adaptive sampling works with a high quality computer on cDNA size stuff. I wouldn't say it works great. I think ideally you would want something at least 8 kb. You could do 5 kb. I would say 5 to 8 kb long. It does technically work on like a 1 to 2 kb size fragment, as long, again, as you have something that can make that decision really effectively. So that's all about your CPU and your GPU. However, the thing that it will absolutely not work on is stuff like cell-free DNA, right? Like we're through the read before we can really consider anything, right? If we're like at 1 to 200 base pairs. We can do depletion. So what we do for depletion is that we upload essentially mostly just like a FASTA, and then we tell the sequencer align to the bed file or the FASTA and reject what you see. So the main use for that is a host depletion. So we see that a lot for example, respiratory metagenomics, or a lot of people sequencing chicken and wanting to know what kind of bacteria is in their chicken. We do find that it works best just with depletion, right? It's that needle in a haystack where there's a lot of hay and not a lot of needle. So a lot of times it works best to pair adaptive sampling depletion with like a eukaryotic enzymatic depletion if you can. And that's really effective. We have a cool poster on it that I'm happy to share. I hope that was helpful as an answer. And then, yes.
Two question. The first one is, so when there's a rejection happening in the pore level, and the strand is leaving the pore, does that like damage the pore at all, or does it like reduce basically how much that pore is capable of sequencing?
So that's a really good question. It's kind of crazy. Like I work with like the Minnow developers, and this is something that like it has taken us like 10 years to perfect. It does not harm the pore. What it does do is when the strand gets kicked out, the helicase molecule tends to disassociate, so it can't come back for a second round, which is advantageous to us. There's a small chance that the pore could be harmed. It's unlikely. What's more likely is you'll have like a small percentage of times where it fails to successfully kick the molecule out of the pore, and in that case, the pore just becomes blocked. So you will notice when you're doing adaptive sampling runs, it's important to have essentially like a well-loaded and conducted library. And by that, I just mean you want lots of molecules so that it has something new to take on as soon as it kicks something out. And you generally want things to be of a good quality so that everything can slip in and out easily. But in general, no, we're pretty good at fine-tuning, and we've had a lot of time to fine-tune the power of the flick, if that makes sense. We spend a lot of time flicking because unblocking long reads is very important to the efficacy of the platform. And there have been times, like I remember like 3 years ago, where we accidentally like cranked the flick power up to like 11, and it was flicking too hard. So but we fixed that. So no, it does not really hurt the pores.
The second question I have is regarding the chemistry 9 versus the R10 chemistry. I understand that the barrel is different. It's contacting longer stretch of DNA or RNA. I've seen papers where the R10 chemistry, even with the same library pack for versus R9, the translocation speed is a little bit slower, and then the average read length is shorter compared to R9 chemistry? Is that because you guys also saw, and then if so, is that because of the longer barrel, or is it not the different mortar enzyme that's attached to the pieces?
So that's definitely a real observation, but it kind of comes from different factors. So sorry, you mentioned shorter read lengths, right? So what's causing, we did see a sort of a dip in read length, and that is not related to the barrel. That is actually related to the introduction of what we call the high capture adapter. So I mentioned the tether molecule, right? It's polyphilic, it's attracted to the surface of the flow cell, and then it's also attracted to the library molecules. We accidentally made it a little too high capture, so it's really, really good at pulling down everything in the library. And we have found that maybe it's a little bit too tilted towards pulling everything down, and that we need to prioritize it or readjust the chemistry so that it's not pulling down small stuff so efficiently. Because what we saw in the switch from R9 to R10 is that we were just getting a lot more short stuff pulled through, which is obviously frustrating because it just kind of drags, it shifts everything sort of to the left. We recommend dealing with that with more stringent size selection, which we know is annoying because it kind of puts the burden on you. That is something that we're sort of tweaking back towards optimizing to longer. So that is a real thing that I've seen for sure. And then the other thing that the longer barrel does impact, right? If you can imagine, and PacBio sort of went through a similar thing. I mentioned them just because like it's interesting to see like what we have both experienced as we push towards like higher performance. As we got the longer barrel, it did like make it harder to achieve really high outputs just because a longer barrel is more likely to get stuff stuck in it, right? It's harder to get unblock from a longer barrel, but we have found overall that it is worth it for the higher accuracy. So there's a little bit of a balance trade-off there where we do better at homopolymer, we have better accuracy overall, but it's a little stickier, if that makes sense. So yeah, I would never tell you like, oh no, there's no difference. But I can absolutely help give like advice on like restoring the type of performance you see on Iron 9 and also why you see like some differences. But I think in general, from the people that I have, there are like a couple applications that people particularly struggled to port onto R10, and one of those was COVID sequencing. And that was because we were using a transposase based kit. So it was like amplicons that were getting cut up really short. And then also we were like feeding them through this new barrel. And so that was kind of rough. But most of my customers have been quite pleased with the R10 change.
Genome sequencing, when you in order to get to the chromosome level assemblies, do you see that you have to combine it with whatever it is?
So one of the things where we're really well advantaged in the field of assembly, and where we have always sort of needed to be included in all of the like big T2T assembly products, is our ultra long sequencing option. So we have a specific kit where we use actually a transposase mediated library prep, but we extract ultra high molecular weight DNA and then add like a very low ratio of transposase to it in order to get very long reads. So like our N50, our average read lengths are like 100 kilobases. So that's incredibly useful, right? For gap filling, for chromosomal assembly. And then, so that's one thing that's been really key for us for a long time. But then on the bioinformatics side, with the AI revolution, there's a couple of error correction tools that, since we're not trying to call variants, right, we're trying to do assembly, they use like an AI directed error correction that has been a huge boon for assemblies, like in like soybeans, for example. Like being in the Midwest, Braun and I do like some agribusiness accounts. And we've gotten super good feedback on that. The question of whether or not you need to do hybrid assemblies for bacteria, absolutely not. A nanopore only assembly will get you there every time, unless you're doing something. There's some like really difficult organisms where
Flies and all that.
flies.
Different species.
I think you can do it nanopore only. When you get into some of like the more complex organisms, you can still do it nanopore only. The question becomes, is it cost effective? There are definitely still some applications where it'll be more cost effective to do a hybrid assembly between like Nanopore and PacBio, or Nanopore and Illumina. But in general, we can achieve like super high quality Nanopore only assemblies, but generally it requires us to do an ultra high molecular weight run. Something we do Pore-C for chromatin accessibility, and then we have like a weird kit called APK that's in development that allows us to do like gap filling and just like more difficult repetitive regions for assembly applications.
Will you be available for discussing specific projects?
Yeah, absolutely. Definitely. Yeah. Yes, we are happy to meet and support, and yeah, anything you guys need. Thank you so much for coming.